Hi, I'm Mr. Williamson. Today's video is about the cell membrane. Make sure you take good notes. If you have questions, put them in Edpuzzle or ask me in class. You can remember, pause the video whenever you need to or rewind it if there's questions or you want to get more of the information in more detail. Let's get started. First of all, we're talking about cell membrane. Sometimes you'll hear about it called a plasma membrane. Basically, it just means that it surrounds the cytoplasm, plasma membrane, cytoplasm. The other thing to remember, like we talked about when we talked about the cells, it's dynamic. It's always changing. It's moving constantly. So it may look the same, and I'm going to give you diagrams and pictures, but it never is exactly the way it looks like in a picture. It's dynamic. It's not static. But that's the way I'm going to present it because that's the only way I know of doing it. The form is a bilipid layer, and I'm going to explain that in much more detail in a few minutes. But it's about 4 to 5 nanometers thick. And I know you're probably like, yeah, what the heck's a nanometer? Well, this shows you kind of what a nanometer is. DNA, you can see down here, DNA is about 10 nanometers thick. So we're talking about less than half of a of a DNA, strand of DNA. You can also see that combustion exhaust over on the other side is about 20 nanometers. So it's about a quarter of the combustion exhaust you see coming out of cars or trucks. All right. So gives you an idea of the size. It consists of proteins and phospholipids. Let me show you what one looks like. This is a diagram of a cross section of a cell membrane. The phospholipid layer is this part right in here. All right, the phospholipids. Each one of these little balls, and I'm going to explain this in real detail. Each one of these balls is the head of a phospholipid, and it has 2 little tails attached to it. All right, and they are arranged in 2 layers. A layer up here and a layer down here. All right. The big blobs that you see, these things, are the proteins, and they can vary. We're going to talk about the functions of some of the proteins in a phospholipid layer. All right. Anyways, but what I'm trying to point out is that they come in a bilipid layer, and they consist of phospholipids and proteins. All right. Moving on. What is a phospholipid? All right, there's 2 parts. Again, do not worry about knowing the structure of a phospholipid. All right, there are 2 parts to a phospholipid. There is a hydrophilic head. All right, this is the part that likes water. It's attracted to water, and I've shown it in 3 different views here. I really like this diagram because it shows in 3 views. It shows you in the chemical formula here. It shows you in a spatial model here. And this is the one that you're going to see most of the time. That's how I'm going to draw it. All right. But you can see there, it's got a phospho. It's a phospholipid. So the word phospho implies that there's a phosphate, and it is right there, phosphate. All right. You can see that there is one saturated lipid, and there is one unsaturated lipid. It doesn't have all the hydrogens attached to it. That should be familiar from last unit. That's why this leg right here bends. Although we don't usually draw that bend, it's there. All right. What we usually draw is this on the far right. A simple circle with maybe a little wavy line or something, 2 little wavy lines. All right. So if you see that, we're talking about a phospholipid. So you've got a hydrophilic head, which is attracted to water, and the hydrophobic hydrocarbon chains, which is repelled by water. All right, which is what gives the bilipid layer its typical view. This is the original. This is what I used to use as a diagram, and you can see the heads here. So you have H2O. All cells have water both inside and outside. So you have H2O there, and you have H2O here. The heads, which are attracted to it, go out. The tails, which don't like it, are kept in the inside. Therefore, they're kept away from that water. They're repelled by the water. So the heads, which are attracted to the water, are pulled towards it. The tails are repelled against the water, away from the water, and they're kept going towards the middle. All right. I like this view better. It's more three-dimensional. You have the hydrophobic tails, which are repelled by the water, so they're going to be hidden inside the bilipid layer. And you have the hydrophilic, the part that likes the water, the heads drawn towards it. All right? Now, you don't need to know this, but I find it interesting. How did they figure out that it's a bilipid layer? Well, this is kind of what they did in a roundabout way. They knew the surface area of cells. We know the surface area of a cube. I'm going to use a cube as an example. If you have a 2 centimeter cube, 2 centimeters by 2 centimeters by 2 centimeters, to find the surface area, you take 2 times 2, multiply it by 6, and you get 24 cubic centimeters. All right. Well, we can do that same thing with red blood cells. We can take the surface area of a red blood cell, and all red blood cells are exactly the same. Human red blood cells are all the same size. Doesn't matter race, color, origin, doesn't matter. Everybody has the same exact same size, shape, and size of red blood cells. All right. That's why we can give blood to one another as long as the blood type and everything are the same, which we're going to talk about a little of that today too. But there's the same size, same shape, and we can figure out the surface area of that red blood cell. So what they did was they took red blood cells and they put them in a centrifuge and spun them out, and they used like chemicals to break them apart until they got just the lipids, the cell membrane. All right. And they put that on water. Well, oil, a lipid, floats on water. So what they figured was that they should have this amount of surface area. Let's pretend that a red blood cell has 24. It shouldn't be cubic. That is wrong. Square. This is absolutely wrong. It should be cubic. That should be square. Sorry, my bad. Should have 24 square centimeters. It doesn't because they're much smaller, but let's pretend. A red blood cell has 24 square centimeters surface area. And when they spread them all out, that's not what they got. What they got, again, wrong, oops, my bad, is 48. They got twice as much surface area as they should have from a red blood cell. So if they had 1,000 red blood cells, and it took up, and they figured out the surface area of a red blood cell is 24 square centimeters, they should have had 24,000 square centimeters. But they didn't. They had 48,000 square centimeters. Why? Because it's a bilipid layer. There are 2 layers of lipids instead of just one, which they thought. They thought it was just 1 layer. They figured out it was 2. And then later on, when we came up with electron microscopes, we could see it in more detail. All right. So what's the function of a cell membrane? Well, one, it maintains the difference between the inside and the outside of the cells, kind of like your skin or like a fence. It keeps what's inside in the cell in. It keeps what's out of the cell out. It's a barrier between the environment and the inside of the cell. All right. It acts as a selective barrier. Here's a diagram of a cell membrane, and hopefully by now you're starting to recognize the bilipid layer here. Okay. And that's the way you're usually going to see it drawn in all the diagrams and pictures. And you can see some things get through it. For instance, small gases, oxygen, carbon dioxide, are able to get into and out of the cell, which is kind of important because we need oxygen to get into the cell for respiration, and we need to get rid of the waste product carbon dioxide when we're done with respiration. You can get hydrophobic molecules. Hydrophobic would mean molecules that are repelled by water. They can get through. Example they have here is benzene. Benzene is not a good chemical. That's why you don't want to be handling certain chemicals like benzene, acetones, things like that with bare hands, bare skin, because they can penetrate through the cell membrane into the cell, causing damage to the cells. We can get small polar molecules in, like ethanol. Ethanol is alcohol or water. What doesn't get through it is large things like glucose. Saccharide does not get through. All right. or charged particles, amino acids, or ions of hydrogen, sodium, chlorine, things like that. So those don't get through. So it's a semi-permeable. Permeable means something can pass through it. So in this case, some things pass through it, some things don't. So it's semi-permeable. All right. Well, I can just take that away. So it's semi-permeable and selective. It actually can select what gets through and what doesn't. We'll get into that later. All right. So the bilipid layer acts as a solvent for the membrane proteins. All right. The result is this dynamic movement. Remember, it's dynamic, not static. Here you can see different protein. And again, this is much more detailed than you really need to know, but it's a good diagram to show it to you. You can see these big egg-like things are proteins. Those are proteins. All right. They're capable of moving through the phospholipids as they're needed. All right. They're not locked into place. They're not like our school where the buildings are put in the ground, cemented, and they don't move. They're more like the students. They can flow. And that gives us this fluid mosaic model. Fluid meaning it moves. Mosaic means it's made of small parts. You made mosaics when you were in kindergarten. You cut up little pieces of tissue paper or construction paper in different colors, and you glued them to another paper, and you made pictures out of that. You know, a lot of, like you made an eye, blue, and whatever. Anyways, that's a mosaic. Mosaic means it's made of real small parts. Fluid means it's able to move. I've got two videos to show you. Very short. This one is not really well done, but it does kind of show you a fluid mosaic model, okay? So you can see they've put the phospholipids, the little balls with the two lipid hydrocarbon chains, and they have a blue protein. And they're just showing you how it can move around. It's not locked into place, all right? All right. Another example, same kind of thing. They've used ping pong balls, straws, and something, and they're showing you how the phospholipids stay in two layers and the protein can move between them. And then finally, I think that this is more of the example. All right. If you look at this animation, you have an individual who is dressed in blue. Think of that person as a protein. All the other people who are dressed in white are phospholipids. The proteins can move through the phospholipids without any problems. They're able to move. The phospholipids will move out of the way. So if we need to move a protein over to another area, it can slide through the phospholipids around the membrane. Okay, so let's talk about those proteins. What is the function of those proteins? Well, one, they're marker proteins. They're like the name badge for your cell. Every one of your cells has proteins in it that identifies it as yours. That's why you can't just give your kidney to anybody. You can only give it to people, if you're donating kidney, that are a match for you, based on blood type, based on the marker proteins. There's proteins that say this cell belongs to this person and it belongs in this part of the body. Your liver has liver cells and it identifies the marker proteins, those proteins on the outside of the membrane say, I belong to this person and I belong in the liver. And if we take those liver cells and if we were to put them somewhere else in your body would go, well, that doesn't belong here, and it would attack it. Okay? That's what happens with autoimmune diseases. The body stops recognizing the marker proteins and it recognizes them as something that shouldn't be there anymore. Okay? Receptors. Receptor proteins are like receptacles, right? They're where if a certain chemical comes along, a hormone or some other chemical, usually a hormone, come along, it plugs into the receptor protein, and that sends a signal across from the outside of the membrane to the inside of the membrane and causes the cell to do something. All right. So if you have, I actually, we're a little bit outside my expertise here. So if you have a growth hormone that it comes in contact with the cell, it'll tell that cell to maybe divide. All right. But it's done through the receptor proteins. And the last one is channel proteins, which just from the name of it, hopefully maybe you can figure it out. It's like a doorway. This is how things get into and out of the cell, right? And they're very specific in some cases. In some cases, just about anything can get through. But they're channel proteins. So you have marker proteins, which are like the name badges for the cell. You have receptors, which transmit signals from the outside of the cell to the inside of the cell. And you have channels, which allows things to pass back and forth. Okay? So, okay, yeah, that hurt. Didn't see that coming. Let's take a quick review. First of all, we talked about phospholipids, all right? They're a hydrophilic head, which is attracted to the water, which is why they're all on the outer layers. And we have the hydrophobic tails, which is not attracted, actually repelled by water. So they're always in the middle. So you're always going to see that, and I sometimes have drawn it just like this, just squiggly lines, and you're going to see the bilipid layer. Okay? We talked about marker proteins, and I'm not sure which of these would be marker proteins. That's, could be this one. I don't know. I don't care. But talk about marker proteins, which is like the name badge. You can have receptor proteins, and I'm not sure if that's an integral, but I'm just giving you an example. It usually goes all the way through the membrane. All right? And then you have the channel proteins, which protein you can see. There's a hole. This is a cutaway. There's a hole for something to go into, or it could go out of the cell. There are other things, and this diagram is actually pretty accurate. There can be cholesterol and things like that, like right here, that are part of the cell membrane, but we're not going to talk about that. All right. Anyways, that's the cell membrane. Hopefully you took good notes. If you have any questions, please feel free to ask in both on Edpuzzle or in the classroom. Good luck. I hope I explained it well. Like I said, if I didn't, ask questions.